Nucleotides get added during DNA replication very, very quickly. So 1,000 nucleotides per second are added to the growing strand in prokaryotes. And in eukaryotes is a little bit slower, but that's still 50 to 100 nucleotides per second. So imagine trying to type out a paper where you hit a button on the keyboard 50 to 100 times per second. Very, very fast. So sometimes typos happen and the base pairs might get mismatched and the wrong one gets paired with the wrong one, etc., etc. And thankfully we have some DNA polymerase that goes in and can kind of help proofread the new strand and fix any typos that may occur. However, proofreading doesn't catch 100% of mistakes. So think about maybe the last time you wrote a paper and you wrote it and then you looked over it and you edited it and you took out the typos and you fixed those and you had a friend look at them and they helped you fix the typos, and then you turn it in, and as you're turning it in you notice that there is a typo that you still missed. That happens. It also happens in DNA replication as well. And when there's a change in the DNA code, it causes something called a mutation. There are other things that can also damage DNA, especially environmental factors. So exposure to different chemicals, radiation, pollutants, these are all things that just over time can damage your DNA. So remember that DNA, which contains your genes, ultimately codes for the production of a protein. So if the DNA code is changed, it may change the protein that's made as an end result. And that change can have a couple of different possible effects. So first of all, the mutation can be a silent mutation, meaning that the change in the DNA doesn't change the protein production, in which case the mutation doesn't affect the protein production and you would never know that this mutation existed. It wouldn't cause any diseases or disorders or any problems. Some mutations can be harmful. So sometimes when the DNA code is changed in a certain way, it can lead to cancer or other genetic disorders. And that's just because the end product, the protein, since the recipe has changed, it's no longer being made the way that it should. So imagine if somebody went into your recipe book and instead of telling you to put in a cup of sugar, it said to put in, 6 cups of sugar. Your cake isn't going to turn out right in the end because your recipe has changed. Some mutations can actually be beneficial, and these beneficial mutations are the basis for evolution. Now, you're not likely to get like X-Men powers and X-ray vision and stuff like that, but if it's a mutation that helps to increase an organism's kind of survival and reproduction, then it's a mutation that's going to get passed on from one generation to another, and it can give the individual some sort of survival advantage. Now, mutations are also more common in an individual over time. So older people are more likely to have mutations in their DNA, and it's one reason why older people are more likely to get cancer as well, because cancer is primarily caused by damage to DNA. So older people, imagine how many times your cells have gone through DNA replication. The older you are, the more DNA replication events your body has gone through, and those typos start to add up over time. So it's kind of a cumulative kind of collection of typos in your DNA, which can lead to more harmful mutations along the way. The older you are, the more that you've been exposed to certain environmental effects as well. So, for example, one thing that can damage your DNA is UV radiation from the sun. and that UV radiation can damage the DNA over time. And so people that were exposed to the sun a lot as a child and throughout their, maybe teen years and young adult years are more likely to develop skin cancer later in their lives. And that's because over time you're exposed to the sun more and more, and these mutations start to build up. Now, we can visualize someone's DNA, we call it a DNA fingerprint, using a technique called gel electrophoresis. And this is, if you've ever watched like CSI or any sort of like forensic crime show, this is one way that they determine whether the DNA at the scene of the crime matches the DNA of, say, a suspect. So what happens is there is a gel called agarose that is made, and it has kind of the consistency of gelatin or jello. So the DNA is extracted. This can either be, maybe blood found at a crime scene, or they can take a sample of cells from somebody that's a suspect. They can do this with a blood sample. They can swab the cheek to collect cheek cells, or some other way to get the DNA. Those DNA, or the DNA that's extracted, is cut into smaller pieces using enzymes that cut the DNA at certain base pair sequences. Those DNA fragments are then loaded into wells, and you can see the wells along the top of the gel here. So this is the agarose gel, and this one has 1, 2, 3, 4, 5, 6, 7, 8, 9 wells. And on this particular one, on either side, on either side, this is called a DNA ladder. So these are DNA segments that we know what the length is, and it kind of helps us to figure out that, if this DNA segment matches this one, then it's about this big. So all the DNA is loaded up here, and then an electrical current is run through the gel. And that electrical current causes the different segments of the chopped up DNA to separate out from each other. And that's because DNA has a negative charge, and so it moves towards the positive end of the current. So remember, opposites attract. And so the DNA gets pulled from the wells at the negative end towards the positive end of the gel. Now, as the DNA is pulled through, those individual segments that were chopped up start to separate out from each other. And that's because smaller fragments can move easier through the gel and can therefore move further or faster, whereas the larger segments tend to get stuck more up towards the wells. So this band here is smaller in size, or fewer base pairs, than this band here. So we end up getting these banding patterns, and you can see how some of them match up. So for example, this one and this one and this one are identical. They all three have the double band that are at about the same area or the same level. So we know that these are matches. So perhaps maybe this was DNA that was found at the scene of a crime, and this is DNA from one of the suspects. Since they match, this would be evidence to support the theory that this person committed the crime. So we can use this gel electrophoresis technique called DNA fingerprinting to, one, do kind of CSI crime scene investigation. So these are bands of DNA. We can see the victim here, that's the victim's DNA, and then this would be a specimen that was maybe at the scene of the crime that doesn't belong to the victim. So maybe this was the victim's blood, and then this was maybe DNA collected from semen. Perhaps it was a rape crime, and so this was other DNA found in or on the victim's body. And then if we had three potential suspects, so maybe it's the um, the victim's, ex-boyfriend or some random guy on the street or, who knows. But wherever the suspects come from, they can collect DNA from them, run it through the same gel electrophoresis process, and you can see that there's a pretty clear match between the specimen found at the scene of the crime and suspect number one. So this could be used as evidence in court to try to get suspect number one convicted of the crime. So if there's a match, it's not 100% proof, but it's pretty strong evidence that suspect number one was at the scene of the crime. Gel electrophoresis, or DNA fingerprinting, can also be used to determine paternity testing. So if we take a look at here, we've got mom's DNA in blue, and her bands lined up here. Dad's DNA in yellow. But you look at daughter number one. So daughter number one is definitely the daughter of mom and dad because she's got some bands that, she has some bands that match up with mom and some that match up with dad. But they all match up with at least one or the other. And that's because her genetics come from her parents. Daughter number two has DNA bands that line up with mom, but her other bands don't line up with dad's bands. So this one is pretty close. This one doesn't line up with mom or dad because it's a little below here and it hits here. So in this case, this daughter number two came from the same mom as daughter number one, but she's got a different dad. If we look at son number one, he is definitely a product of mom and dad because once again his bands are similar to the first daughter's bands in that he either has bands that match up with dad or bands that match up with mom. So he gets his DNA either from dad or from mom. Now, son number two had to have been adopted because his bands don't match up with either mom or dad. So he's got these bands that kind of hit in totally different areas. And so son number two would have been the product of a different man and a different woman having a baby. And if he's part of this family, then he could have been adopted.